And so it's a great pleasure to introduce Stephanie Portet from the University of Manitoba in Canada. And she has been doing a lot of work on modeling filaments, microtubules and intermediate filaments. And she's going to speak about modeling intermediate filaments.
Okay. Thank you for the invitation and good morning. Still morning. Okay, yeah, so cytoskeleton. Cytoskeleton is made of three types of filaments: the actin, the microtubule, and also the intermediate filament. So you have in here 4 cells. In blue you have the actin, in green the microtubule, and in red the intermediate filament. So you have the 3 different channels in here. So different architecture, different function. So for the actin, a lot of people speak about that. So stress fiber and in a specific organization in the cortex. For the microtubule, you're going to have more a radial organization. And for the intermediate filament, my topic today, we are going to see. So we had a good introduction yesterday by Rudolf. So I have just now a very short summary, what we have to remember. So first, there are tissue dependent. So you're going to have keratin in epithelial cell, vimentin, desmin in muscle cell, and so on. So you have different types, but all the time you are going to have the same shape for the subunit, something elongated. It's not globular, and they are going to form a cable and almost about 10 nanometer of diameter, but we are going to go back to that. So that about for the structure, for the other type of specificity. So we have seen yesterday they are non-polar. They can, when you put purified protein in a tube and no ATP or no GDP, they are going to assemble. They are regulated by post-translational modification. Yes, phosphorylation for intermediate filament is very important, and the network is shaped not by other type of protein, but more than this modification. That's very specific mechanical property. For example, they are mainly elastic, so you can pull up to 3 times to the resting length, and they are not going to break. Okay? And related to this property, you have some function, and so they are mainly involved in structural function, but also in functional behavior, so like the cell signaling. But in here, what I'm going to use is the epithelium as an example to illustrate the structural, function of the intermediate filament network. So you have an epithelium, layer of epithelial cell, and in the epithelial cell, what you are going to have is a keratin network that is going to span the cytoplasm. And we are going to connect each cell by desmosome, so we are going to provide not only the mechanical integrity of the cell, but also of the tissue. And now if we keep going with this epithelium example, but we take the a pathology, a disease as an example, the skin fragility disease, EBS, and this disease has been linked to a mutation on the gene expressing the keratin. So if you take cell with normal keratin, you pull on your cell, what is going to happen? So the network of intermediate filament is going to deform, absorb the stress, and the cell and the tissue are going to be preserved, let's say, maintained. If you do exactly, so in here you have the normal cell, the diagram equivalent to that. Now if you do exactly the same thing, but on cell having this mutation related to EBS, what you are going to observe after a while is the network is not organized anymore as normally, but you are going to have the protein aggregate in small particle, aggregate, I don't know how you want to, granule, how you want to call that. And so finally, so now in this case, the mechanical integrity of the cell is not preserved anymore, but neither the tissue. So what you are going to observe at the level of the tissue is going to be some detachment of some layer, and so you have some pathology. In here, the main problem is we do not have anymore the proper organization to maintain the function. Okay? But the problem is where this thing comes from. The mutation is inducing a fragility of the filament, or it's just the assembly process that is going to be defective. We don't know. But what is very important in here is this link between structure and function, and the organization of intermediate filament are going to provide help them determine the function. It's for this reason I'm going to be interested in the organization of the network, and I'm going to try to understand finally what was my question, the contribution, what are the contributors of to the organization of a cytoskeleton network in general, but more specifically and to the intermediate filament. So in here I'm going to present 3 work, and all the time I have data. I'm just a mathematician, nothing else. So the data are not coming from me, okay, or from my collaborator. And so the first question here was, before organizing the network, we need to elongate a filament. So how a filament can be organized for the intermediate filament. This work has been done in collaboration with some lab in Heidelberg. And so what they do, they put, so in here we are going to work in vitro and with vimentin. Vimentin is from a mesenchymal cell. So what they do is they purify the vimentin, and they put the vimentin and they wait for some time. And what we are going to observe step by step is the elongation of filament. What we are going to do, we are going to measure the length of the filament. So we are going to have the mean length as a time variable, and also the distribution of length. So for the first work, we are going to use this data, the mean length. In the second work, we will use the distribution of length. So first, we need to build the model. So according to what we observe for sometimes, so remember, Rudolf was speaking about the tetramer. So the tetramer are going to be elongated subunit. And so if you want to imagine, like that is about 40 nano, and that is about 2 nano. So first we are going to have the tetramer, the T's that are going to laterally aggregate to form an octamer, and after 2 octamers form hexamer, and after finally what we call the ULF, unit length filament. And the unit length filament are going to be the building block for the filament. This thing has about the same diameter as the filament at the end. They might have some compaction sometimes when the filament gets mature, but this thing has about the same diameter of a filament. So that is just to build the ULF. And notice we do use exactly the same constant of association for this lateral aggregation. After that, we need, in this work, we assume a initiation of filament. So to initiate a filament, we use now this longitudinal annealing of 2 ULF. And now, finally, the question, how to elongate a filament. So we assume 3 possibilities. So longitudinal annealing of a tetramer to the end of a filament, filament plus filament longitudinal annealing, or a filament to a filament. So we are going to have 3 constants. So Tk to represent the tetramer addition, Ku to represent the filament ULF, and the filament longitudinal annealing is going to be represented by KF. And so we have our complete model, but to compare that to the data, we have formed an expression for the mean length, so as a dependent variable, so time-dependent variable. And in here, we have just the solution of our dynamical system. Now, what we do is we need to, so remember, we have three possibilities, and we are going to examine all the possible scenarios. When we are going to have tetramer addition and ULF and filament, and so on. So we have all the time, we are going to switch on and off the three possibilities, so 2 to the power 3, 8 scenario, and we are going to compare the 8th scenario using the mean length to the data. So so what we do now, we have our 8th scenario, we have some coefficient in the system, we are going to fit the data, have the best thing possible, and we are going to compare the behavior. So now, right away, remember, we start with 8 scenarios, but we have only 4 responses. In fact, the first thing is we do not have any influence about having or not the addition of the tetramer, because the tetramer finally are all consumed in the aggregation of the ULF. So there is nothing left after a while, and this aggregation, this lateral aggregation, is very fast. It's going to happen in the 2-3 seconds in the aggregation. So first, when we have just the tetramer, so nothing happened, we just form the ULF. When you have just ULF, we are going to have just the initiation of filament, nothing else. And after, when we have just the filament, it's not too bad, we can elongate, but we have a problem in here, and in here we have both filament addition and ULF addition. So now, when we are really comparing everything to the data, we find the best scenarios. The best scenario are when we have the ULF and the end-to-end filament annealing. And what is very important is to elongate any filament, we require this filament addition. If we do not have this filament addition, we cannot elongate properly the filament. And so is what we were trying to propose in 2007. So first we have this first phase, the lateral aggregation to form the ULF, very fast at the beginning, and after you have the ULF addition and also the filament addition. So at the beginning, to try to publish this paper was quite difficult because this guy, a lot of people were saying, no, that cannot happen. Fine. But 2 years later, the biology, now, different group, okay, from Chicago now. So what they have done, they take so in some cells they label, and it's exactly the same protein, it's vimentin 2. So they take the label in red in some cell, in green in some cell. They mix, so they fuse the 2 cells, and what they observe at the end is this annealing of 2 filaments. So this is really happening. They have this ability to anneal. Okay? So in this first work, we use only the mean length. Now, what about the other thing in the data? We have also the distribution of length. So in this case, what we have, mathematically speaking, is a coagulation fragmentation problem. I forgot to mention before, when you put the protein in vitro, what the intermediate filament protein are going to do is they are going to aggregate. they do not observe any disassembly. We have disassembly in vivo, in the cell, but because there is some phosphorylation event. Okay? Without the phosphorylation or without any other post-translational modification, you do not observe disassembly. So now, in fact, for us, it's going to be just coagulation. But so the tools, the coagulation fragmentation equation, you are just going to follow the concentration, the amount of some object of a given size. And that is just the continuous and infinite version. So you are going to have contribution of how, so the formation of the crust of the object of size x due to the aggregation. Otherwise, you can also lose some object because they are going to break up. Otherwise, you can also lose some object because they are going to be involved in the formation of bigger object. Or you are also going to create object of size x because they are going to be, bigger objects are going to fragment. In here, we can simplify a little bit, a lot, this problem. Why? So first, we forget about the fragmentation, that is the worst finally. And we forget about the fragmentation because we do not have any disassembly or severing. In vitro. If I was working in vivo in the cell, it was not working. After, I maybe did not mention that before, but I can have the mean length, the length of the filament, not like in nanometer or in micrometer, but in number of ULF. Even there is interdigitation, we know exactly how much, we can calculate how much a ULF is going to contribute to the length of filament. So the ULF is 60 nano, but it's going to contribute due to the interdigitation only to 40 nano. So instead of having the length of filament in nanometer or micrometer, we are just going to count, so we in number of ULF. So we are going to have discrete in size, so not anymore continuous in size for the coagulation equation. And finally, also what we have is a closed system. We are not going to degrade and synthesize any other protein. So we have a closed system, a finite version of the equation. In here also, I'm going to forget about the formation finally of the ULF, because that is done in the first second of the dynamic. So what I'm going to play with is only with ULF and how the ULF are going to aggregate to have a filament of longer 2 of length 2, my French is coming back, and or elongate the filament. So we have, for the variable F1 is just going to be the concentration of filament of length 1, so the ULF. Otherwise, we are going to have the concentration of filament of length I. And what we are going to play with is finally the constant of association, and we are going to assume they are going to be dependent on length and also the shape of the object. So we are going to have this type of equation. So we have n equation, n being normally the total number of ULF we put in, but we can have a smaller number of equations. We are going to have, so the constant of association, that is going to be the big thing for us. And we are going to consider this equation with the initial condition at the beginning. As for the experimental protocol, at the beginning we have only ULF. And so now, what about this KIG the constant of association between a filament of length I and a filament of length G. So first, before aggregating, they need to collide. And to describe the rate of collision and if our objects are just moving around due to the diffusion or kind of Brownian motion, we can use this expression from Smolichowski. So in here, what you are going to have is the diffusion coefficient of the first object, the diffusion coefficient of the second object, and what we call the interaction radius. If two objects are too far, no way they can collide. And now the problem is how to describe this quantity, the diffusion coefficient and the radius of interaction. For that, you need to, you need the size of the object, but also you need to know what type of description you take for the object. So you can use, for example, a flexible micromolecule, or you can imagine something totally rigid. Depending on the description, you are going to obtain different expression for the diffusion coefficient and the radius of interaction. Yes.
So that means you are working here in dilute solution. We don't have entanglement effects or stuff like that.
So again, sorry.
Oh, sorry. So we don't have entanglement or network effects. It's dilute solution, I guess.
You remember so we have no network. No network at all. Yes. No. This model will not work if we have a network. The filaments are single, not connected. Yeah, good point. Yes.
Another question. I mean, a rod has a diffusion.
Wait, I don't. Sorry.
Okay. So there is actually a tensor of diffusion constants. You both have translational and rotational diffusion.
No.
And you actually have a diffusional tensor, not a diffusion constant.
I'm fine.
See, there's longitudinal and transversal diffusion here.
Okay, for the expression of the diffusion, when I take a rigid description, I just use the Stokes-Einstein equation, and for the drag, I'm using the description of an elongated cylinder. Okay, and that comes from this paper. And after, for the semi-flexible object, I use this expression for the diffusion coefficient coming from the semi-flexible theory. We can have all the details. I can give you the reference if you want. And for the interaction radius, for the rigid, when I assume uncooked spaghetti, finally, it just the radius, the sum of the radius of the two objects. And after, for this thing, in the case of the semi-flexible object, I just use the mean square of the end-to-end distance. Okay, so that is depending on if I want to have a cooked spaghetti or uncooked spaghetti to describe my filament. And after, okay, so for now, what we have is only the collision rate. But when we have collision, after is not necessary, we are going to have aggregation. So to have really the constant of association, we need to multiply the collision rate by a probability. And what is the form of this probability? I don't know. So I assume two things. First, the probability of aggregation given we have a collision is dependent on the length of the filament, or it's just constant. So whatever the length of the filament interacting is going to be the same. And in here, in fact, the probability is just inversely proportional to the surface of the sphere, including the two objects. Okay? So now what I have is I can form different expression for my KG. My KG is rate of collision times probability of aggregation. So two types of description, rigid object or semi-flexible, and after space-dependent probability or not space-dependent, sorry, length-dependent, depending on the length of my object, or just constant. So I'm going to form 4 model again. And now if I look a little bit more closer to the shape of my coefficient, I just take the coefficient for my model 1. Model 1 is when I assume I have just a rod-like object, and we're going to just interact at the end. And so in this case, I'm going to have for my KIG the constant of association, this type of thing. But what is important in here is I have one part that I can calculate explicitly that are going to depend on the length of the first object, the length of the second object, the diameter of my cylinder, the viscosity of the system, and the temperature. And I have something else. I have no clue what it is. But all the time, for my 4 expression, for my 4 model, I can decouple my association rate in 2 parts. Something that I can explicitly calculate, and a constant. And this constant is going to be the same for a given model, is going to be the same for all the KG. So finally, what I can do is my K0 go out, and what I will have is this DIG are going to shape my distribution, and this little guy is going to tune how fast I'm going to go. And I'm going to compare my 4 model to identify, to estimate this K0 with the data at each time. So I have several times. I have 1 minute to 20 minutes, and after 2 hours and 4 hours. So what we observe now is up to 20 hours, the model having a size-dependent probability are going to represent pretty well the data. However, we start to see a little problem in here when we have longer filament that totally miss that. And the model with the constant probability of aggregation, they fail totally. However, when we look at the distribution of length at 2 and 4 hours, we have a switch. Now, the models that are going to have a constant probability of aggregation are going to really represent the problem. So now we have 4 models, but nobody really represents the time, all the dynamic of the filament, so we have a problem. After looking for long hours at the rate, I cook a little mathematical expression that provide me some rate, that if I use this rate, I'm going to have a good fit of all my time scale from 1 minute to 4 hours. Okay, but I don't care finally. Now I have something working. But what is interesting now is what are the shape of the rate? And can I learn something from this rate? When I look at the rate giving a good representation of my total dynamic from 1 minute to 4 hours, what I'm going to observe is the rate of aggregation of short filament. I'm going to define short filament after, and it's not my definition, it's something appearing from here. I'm going to have a size dependence on the rate. When I have longer filament, the dependence on the size is going to fade. And in fact, what we observe when we look at the rate, we have a kind of change of strategy for the rate of association. okay, 12 ULF, but what is 12 ULF is the half of the persistent length. So in fact, when we go back a little bit to our dynamic, so remember at the beginning I was just speaking about, okay, 1 minute, 5, 10, 20, and so that is a dynamic, so where I have my distribution. And instead of looking at that as like time point, but I look at what I have in my system finally at 1 minute and 4 hours. It's totally different. And so when I look at what I have up to 20 minutes, in here in my system I have at most 50% of my filament larger, so the median length of the filament is 1/3 of the persistent length. So in fact, up to 20 minutes, my filaments are very short. What I have in the system at 2 and 4 hours is totally different. In here, we have the median length about the persistent length, and in here, 1.5 the persistent length. So it's for this reason, in fact, at the beginning, when I was trying to fit, so the rigid approximation is going to work at the beginning, up to 20 minutes, but I'm failing after that, and so I need the flexible representation. So, in short, what I have in here is the importance of the flexible nature of the filament, even in the aggregation and the process aggregation. So if you want to remember something for the in vitro assembly of intermediate filaments, so in here it's going to be more what we call type 3, so it's going to be Vimentin, Desmin, and yeah, there is other thing, but
Keratin.
no, keratin is going to be more. We have heterodimer, so I don't know if it's and keratin also they have assembly. They do that and that in the same time. In here they are just doing lateral first, longitudinal second, and I think in keratin they mix a little bit. But for Vimentin and Desmin, at least for now, we can represent the process of aggregation like that. So first, at the beginning, lateral aggregation formation of the ULF, and after ULF, filament, 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 and we have an important role of the flexible nature of the intermediate filament in the process of assembly. Now, if we look at something more complicated, as shown by Rudolf yesterday, if we want to play in vivo now in the cell, it's going to be more complicated. And now we are playing with keratin in epithelial cells. So if we use in the next work the data explained yesterday and published in a PNS 2 years ago by Rudolf and his group, we are going to use this data. So we have the repartition of the spatial repartition of the keratin. We have the speed. We have the direction of speed, and also we have some preferential area of aggregation and disassembly. And in here we are going to try to see what are the effects of turnover and transport. So for this work we use this data. So we are going to have the distribution of keratin on a diameter. So we have one membrane here and here, the center of the cell. The profile are at 24 hours and 48 hours. So that are going to be the data we are going to use, plus the speed and also the preferential area of assembly and disassembly. And so now, if we want to form a very simple and macroscopic, at a macroscopic level model to try to understand what are the interplay between the reaction and the transport. Okay? So in here, we are going to have only two variables, the soluble and the insoluble. The insoluble pool is just all the keratin protein aggregate into a particle, filament, network, anything they can visualize by microscope. So we are going to have, for the soluble and the insoluble, the equation with the transport and the turnover. So assembly, disassembly, and so we are switching off and on. And for the initial condition, when you have intermediate filament, almost always, so 95% of the material is aggregate all the time. Yeah, except when they already dissolve everything to go for mitosis. And for the boundary condition, we have no flow, nothing go out at the membrane. Now, for the transport, again, guess what? I'm going to make different scenarios to compare the scenario to see what are the best scenario possible to represent the data. So, for the soluble pool, what I'm going to have, just diffusion. For the insoluble pool, what I will have is a very small diffusion that is going to represent this jiggling we can observe in the movie. Otherwise, also, I'm going to have, remember the data, we have this inward motion. This inward motion are due to the transport of intermediate filament along the microtubule due to, in this case, is going to be denailing. But also, in some other case, you could have association of intermediate filament with kinesin, and so in this case, we are going to go on the other way. But in here, seeing the data, we put only this inward motion. We have two types of speed, because first we have the speed from the data, and after we use also, okay, in here, so the speed from the data, in here, that is measured from experimental data. Otherwise, the second speed we assume is just the average speed of the data. In all the time, we are going to have something about 0 at the nucleus. Because if we assume we are going to follow the microtubule, and if the microtubule are just around the nucleus, we are just stopping around, we have about no motion, let's say, around the nucleus. So we are going to have three types of transport. Now for the turnover. We have a very macroscopic model. At the beginning, we assume just a linear exchange between the soluble and the insoluble pool. So we have just some rate proportional. So for the assembly, we are just proportional to the soluble pool, the disassembly proportional to the insoluble pool. And also, what we assume is a second turnover option, a nonlinear option now. We are going to have, for the disassembly, enzymatic effect, so that is very well documented. We have phosphorylation by kinase to induce the disassembly of intermediate filament. And after, for the assembly, we can also, we don't know, we might have some chaperone, or also, in fact, what is going on is, so you have phosphorylation of the aggregate proteins that are going to induce the solubilization. When they are soluble, they are still phosphorylated, and in here, maybe before being able to reorganize in filament, they need to be dephosphorylated. And so in here we can have another enzyme acting. It's for this.
What is the boundary between soluble and insoluble?
What do you mean boundary?
Well, is it a tetramer or a ULF that you're calling soluble?
Alors, soluble, insoluble is something we are going to be able to detect with the microscopy. It's because it's what I'm going to compare to the fluorescent profile. So it's going to be big enough. So the soluble will be tetramer, will be smaller than
Filaments as well.
Yeah. And after, so in the literature, what they say, the soluble pool might be tetramer in vivo. Rudolf?
Yeah, it's a common belief. But we can't distinguish between a single tetramer and a single ULF and multiple ULFs at this point.
Yeah, it's too
It's
small
somewhere
to
in
see.
between
So.
there.
But in this model, the insoluble is what I'm going to be able to, what is able to be detected by the microscopy. Yes, okay, so we have, oh my goodness, we have two types of turnover, and in here, as you notice, we can have constant rate or space dependent. Space dependent, why? Because from the data, they observe some preferential area of assembly and some of disassembly. So we wanted to take that into account. So it's for this reason, for the rate of assembly, we assume a first possibility, okay, everything is constant all over the cell, or we take exactly the same shape as the source region from the data. In here, again, we are going to have some fitting to do to identify, to estimate what is the level of this guy and what is the level of this peak. But the shape is not going to change. The shape is obtained from the data. Again, we have exactly the same type of idea for the rate of disassembly. So something constant all over the cell. So we use also the shape of the sink from the data. Also, we assume a third possibility. We are going to have the disassembly mainly focus around the nucleus. And again, we need to find what is the level of this guy, and so on. So now, if I go back, now I don't have 4 scenarios, but 36. So I'm going to have diffusion all the time, very small for the jiggling of the filament. For the transport of the insoluble, I'm going to have this inward motion, constant or non-constant speed. After, for the turnover, I'm going to have linear exchange between the soluble or insoluble pool or the enzymatic effect. And after, for the assembly, I could have localized or non-localized all the time. And for the disassembly, non-localized at the sink or in the middle of around the nucleus. So finally, I form 36 scenarios. Okay, so first we need to estimate the parameter of all the scenarios. When we have a linear model for the turnover, we have only 2 parameters. When we have an enzymatic effect, we have 4 parameters. We estimate everything using a very basic thing, and for the minimization, some genetic algorithm, so we need to wait some time. So we get the best fit, but now the big problem is try to understand what is the best scenario, and we need to take into account the goodness of the fit, but not only, we need also to take into account the number of parameters, because in the linear turnover we have only 2 parameters, and in the nonlinear turnover we have 4 parameters. So we use this archaic information criterion to compare the model. And also what is interesting, we can assign at the 36th scenario a probability that is going to identify how likely to occur, how a scenario is likely to occur, taking into account the data we have and the collection of model we use, because we compare everything to the best scenario. So in here we are going to be able to rank our 36 scenario and assign to the 36 scenario a probability. And this way we can group our scenario with, okay, what type of transport I have. If I have only passive transport, only diffusion, what is going on? If I have active transport but with constant speed, what is going on? And so on and so on. And so this way I'm going to be able to extract what are the more likely process to occur. So in this case, so when we have, for the mode of transport, when I'm comparing all my scenario, what is emerging is the active transport is required. What is very is good in here when I'm comparing the model, so I put all my model with a linear turnover, all my model with a non-linear turnover. I sum the probability of each, and what I'm going to have is a very larger probability to have the enzymatic activity to occur. That is a good thing, because the first, the linear assumption was totally unrealistic. And so this way we have some, it's good to see the approach is reacting well to detect something like, say, we can expect. For now, the assembly and the disassembly, I'm going to speak about the best scenario, what is in the best scenario. The best scenario in this case, and the best scenario have a very high probability in comparison to the other one to occur. So we have constant drift. We are going to have a constant assembly all over the cell. The disassembly is going to be localized around the nucleus and, okay, enzymatic activity. This model preserves the 85 all over the time, the 85% of material aggregate insoluble. That is good. And what is also interesting is this best scenario is in a good agreement with what has been proposed in 2011 by the group of Rudolph. And finally also, we can look at this best scenario, and when we compare the time scale and so on, we have first for the transport, the advection is dominating, and after if we compare transport against reaction, we are going to have the advection, the main process. Thank you.
So it seems that, so I have a comment, I guess, and then a question, that in your list of scenarios, each and every time the more complicated scenario is the one that fits best the data.
No, it's not.
Well, but the first one. So if you go back to your previous page, it's a catastrophe. It's
What, for this work or?
no, yeah, just one slide earlier or two.
Wait.
No, back. One more. One more. One more.
There is
the
no
other
more.
direction.
Ah,
So, no, back. So, beside the first one, it's the knowing exchange that is more likely than in exchange. Like was
No, yeah, no.
That's a assembly. It's the assembly, localized assembly. It's from a curve fitting point of view, it's a nightmare because it suggests that. I mean, I'm not saying that. So I'm always like modeling when the more simple scenario is what explains the biology, and not like the more complicated one. Then I'm trying to understand what's the value of modeling. And I have to put all that out there. You know, I just have to. You know, if it's all just the more complicated scenario, what does that mean then?
no, it's not necessarily the most complicated. My approach in general, I'm very uneasy to just say, okay, that is going to work like that. It's for this reason all the time what I try to do is take a lot of hypotheses, form the scenario, and I'm trying to go blind. That is my point.
You did. And it turns out, at the end of the day, doing all that, which is unfortunately not done enough. You've done something really nice, and for instance, finding out what's the more likely scenarios through this nice analysis. I really like it. That is clear.
But no, but
I'm
if
trying-
the more
outcome turns
complicated,
out is that what works is the more complicated.
no.
scenario.
The more complicated will be, we have, okay, wait,
I mean,
no.
the knowing exchange with
No.
parameters.
The more complicated will be, we will have non-constant drift. We will have
Wait,
a
back.
non-constant.
Your constant drift, the more simple scenario is no drift, right? So that's more complicated than just, right? There's
But it's simpler
variable.
than variable drift.
But it's
As
not
I
the
said,
most
that was the
complicated.
first case which I skipped. The next three is basically the more almost complicated scenario that wins over.
It's also
Not necessary.
You have to have, I mean, you basically cooked up a disassembly localized around nucleus. Sure, I mean, that's what the cartoon.
Yeah, but okay,
But
my
Lynn was
point
saying-
now is, okay, so now I need somebody in the lab try to tell me if this is stupid or not.
But okay, I guess what I'm
That
trying
is just
to
a
get at-
proposition, and I'm not going to say it's true. What I need now is somebody try to explain why this assembly, why and how the disassembly could be localized around the nucleus. That is something we need to do that in the lab.
Well, yeah, that's validation of your model when you actually go back and verify that you have prediction and that the model is predictive power.
Yeah.
But I'm just concerned that overall you do not simplify. You even added enzymatic activity just as a, and you kind of, I know it why you did it, right? You put this localized disassembly around the nucleus because the other scenario, even intuitively wouldn't work. So you added it there, but that's all made up, right? I mean, I can come up with another 25 scenarios.
Yeah, but
Then
but
when
what
you
are we
stop,
doing when we are doing a model?
Simplify.
Yeah.
Come up with the fundamental. That's just, I don't know. So that's one thing, I guess. The second thing is a question. When the skin is subjected to shear forces,
Yeah.
do you know where it breaks down? Is it through the cytoplasm or is that at cell-cell junction?
That I don't know.
And
Cytoplasm. It's in the cytoplasm.
Yep.
True
So-
cytolysis.
So the junctions remain mostly intact.
Absolutely, yes.
Yeah.
Okay, that's important in that then it's more about indeed the network and,
Yeah.
and less about those junctions.
Yeah. Okay, wait.
Oh, I can shout.
Okay.
You spent a lot of time at the beginning showing us this detailed model of the length distribution, etc. And then when you came to the last part, you threw that away. How important is that? How do you convince yourself that you can ignore all the length effects?
No, the problem is if I want to have the length, I need the data. For the moment, the data I have, I cannot play at this level. That is, it's for this reason. It's for this reason this model is at the microscopic, you have to adapt to the data.
But no, to connect the first to the second model, that's what you have. To connect the first part of your talk to the second part.
Yeah?
Integrate those two.
Yeah, the first and the second are connected, but not the third.
I don't. There was a third. But
So how you ask how can I include the length in this one, in the in vivo?
Let me phrase it a different way. If I were an experimentalist and I knew I had all these other processes going on, and you come up with a model that says, okay, disassembly happens here, please go out and check it, would I invest a lot of time in that process? How do you convince me to do that?
To try to.
Rudolph, are you convinced?
I don't know.
I'm not, I'm just,
But
I'm
I
trying
think
to play
you
a
were
good
talking
devil's
about the first and second part, the in vitro to in vivo part, and I think this is a really important question that I'd be extremely interested in, is does this in vitro assembly occur in the same fashion in a living cell, and can we maybe use this modeling to test images that we get now maybe with super resolution imaging or something, that we can actually probe this model. And that would be interesting because nobody really knows that.
Okay.
Okay, any further questions at this point? If not, then let's thank Stephanie again.